Just starting again because of the recording. Hello, everyone. Thank you for being here with us today for another Solanum seminars. We are happy to present to you today Ikram Bashir. That's going to be our speaker. And Ikram is, just one second. Ikram is native from Pakistan, and he completed his master's in the University of Agriculture of Faisalabad. And currently he's a TWAS NPQ fellowship, where he's pursuing a PhD in the Federal University of Pelotas in Southern Brazil. And his main research focus is to deal with the evaluation and introgression of desirable traits of wild potatoes for the breeding of the cultivated potato. Okay. So Ikram, can you hear me well?
Yes, I can hear you.
Okay, great. So thank you for accepting the invitation and for presenting to us. If you're ready, you can share your screen.
Okay. Okay, can you see my screen?
Yes, you can go to full mode.
Okay, it's good.
Yes, perfect. I can see you, I can hear you. So be our guest, please, whenever you're ready.
Yes. Thank you for inviting me to present in this whole seminar. So as Leonardo already introduced me, I'm Ikram Bashir, doing PhD from Federal University of Pelotas, and I am conducting my research at Embrapa, which is the Brazilian company for agriculture research. So first, I would like to introduce my group, which is Hidden Genetic Resources Lab. So in this group, we are mainly focusing on the potato, sweet potato, and rice crop wild relatives. And we are doing abiotic. We are mainly doing abiotic screening of these crops and try to introgress these desirable traits into cultivated potatoes. So at Embrapa, Clima Temperado, which is based in the Pelotas, we have a potato gene bank, and this potato gene bank is comprised of almost 489 accessions of potato, in which 144 are directive collection from field, and and we are missing information of 5 accessions, and 177 are introduced from other research stations, from other gene banks, and from other breeding programs from within the country and from the USA also. And 163 are the results of the breeding process. And among these, 134 are tetraploid cultivars, 118 are the wild cultivars, and 83 are the soma-colonals. Among the wild sessions, we have almost 78 Solanum commersonii, and 36 we have the chacoense entries, and 336 are from tuberosum, and 26 we have Solanum malmeanum. So after collection or introducing from other introducing the germplasm into our gene bank, we conserve them in 2 ways: in vitro, and in greenhouse we multiply these accessions to get more tubers, and to use them in experiments, and to give them in exchange, and other activities. So the germ plants which we collected here at this gene bank is mostly from the southern part, south part of the Brazil, which is the mostly we collected germplasm in Rio Grande do Sul. one of the state of the Brazil, and a few of the germplasm. We conduct activities of germplasm collection in Santa Catarina, Paraná, and one entry we also get from the Conversely. And here, as you can see, that the Solanum commersonii is the number of Solanum commersonii is the most we collected in the Rio Grande do Sul. Almost the 80-85% Solanum commersonii as we have in our germplasm bank. And these are the 4 main states from where we collected most of the germplasm. So now how we are going using this germplasm, and why we have to use this germplasm, potato germplasm. So for that we have to look at the background of the potato. What is the importance of potato? As from many studies have reported that potato is of almost 1 billion people diet. So until 2050, the world population is going to grow more. And so we need to have more food. For that, we have to increase our production. So currently, until from the last year data, 2021, China is the biggest producer, followed by India and and other countries. So we still need to increase our production. So what should be the idiotype of potato plant in the changing condition, the changing climatic changes, conditional climate, and in the climate, in this changing climate? So here I listed some of the key impacts of heat and drought stresses, which is the main abiotic stresses affecting the crop plant most. So the idiotype should be the higher early maturing varieties. We should have early maturing varieties with this early closure of canopy, so that it can ensure the maximum photosynthesis rate, and the available water should be the available and uptake of available water should be higher, so that it can fix more carbon and from available water, and increase the photosynthesis, and produce in the last, so it can produce more tubers. And what if we don't do this? So there is a study of Hidman who proposed that if we don't adapt to the changing condition of the climate change, then if we don't do the adaptation to these scenarios, then we can lose 18 to 32% of our crop yield. And even if we do the adaptations, because this is the climate change, the continuous process, so then even then we can we will lose a 9 to 18% of population of our yield. So so we have to develop a strategy to minimize these losses to almost 50%. For that, last year we published a chapter in which we focus on the importance of the wild potatoes as a genetic reservoir for potato breeding. In this, we discuss almost all the potato species, and their possible role in potato breeding for to adapt in the drought and heat stress conditions. In this slide, I discuss about the work we are doing at Embrapa. The germplasm available here, we select randomly. I have screened almost 44 accessions, out of which 24 are belong from Solanum chacoense and 15 from Solanum commersonii, and 4 are the result of the breeding program from the university. So we use these accessions and we conduct heat screening. In heat screening experiment, we have a facility of phenotyping, in which we have growth chambers in which we can control the temperature, humidity, and light intensity. And but we have to do manually irrigation. In this chambers, the chamber one we assigned as control, and second chamber we kept as heat stress. We apply stress. In control, we give a temperature range of 14 to 27 degrees centigrade, but in heat stress condition, we give a temperature of 24 to 34 degree centigrade. And we kept 50 to 65% humidity throughout the day. We have a 12-hour photo period, and the light intensity was not more than 400. And daily irrigation was done so that we can avoid any negative effect of drought. So we can have better result only for heat screening. So in the heat screening, we mainly focus on the two type of physiological analysis. One we used LICOR 6400 XT system, through which we get data for net photosynthesis rate, stomatal conductance, and on transpiration rate, and on intracellular and ambient CO2 assimilations. And we also used imaging PMM M series for chlorofluorescence analysis. So before doing these two analyses, we always kept the both chambers at a constant temperature. Like in control, we kept the temperature at 24 degrees centigrade, the temperature 24. But in heat stress condition, we maintain the temperature at 34 degrees centigrade. And and also if while doing chlorofluorescence, we turned off lights before collecting leaf samples to perform fluorescence analysis, and we kept until the end of this analysis. So with the in chlorofluorescence analysis, we mainly focus I mainly focus on the five different mechanisms: the FV over FM ratio, which is a well-known stress indicator, which tells us about the photo inhibition of photosystem 2, and then we move toward the catching analysis, in which we measure the effective photochemical quantum yield, coefficient of photochemical fluorescence, which tells us the open reaction center, and ETR, electron transport rates. And also in the end, we also analyze the non-photochemical catching, fluorescence catching, which tells us about the which gives us about the information of the photoprotectant mechanism. So we also in heat screening, we also performed some made some agronomical traits. In this regard, we published in potato research, and we measured agronomical traits such as shoot weight, dry shoot weight, big tubers, small tubers, weight of big tubers, and dry matter content. Although we don't have any pedigree information, or we don't have any populations, so but still by using mixed model methodology like REML blob, model number 54, we use this, and with this model, it gives us the information on the heritability and CV and other information important for the selection of genotypes. So this the beauty of this methodology is also it ranks the genotypes by their true genotypic values. So true genotypic values. So with this under heat stress condition, from ranking recommended by this model, it is just us that the genotype dihaploid BGB 88 and chacoense genotype BGB 113, they are in the top of the list in according to the genotypic values. And so we can say that these genotypes we can select, and we can move them to our breeding for more analysis and for more look deep into these accessions. And other analysis we perform on the physiological traits in which I perform use the PCA and clustering technique. And from the mean analysis, it suggests that mean analysis suggests that under stress conditions, in a stress condition, the photosynthesis rates were increased by applying stress on the because as we measure the different rates on different time intervals like 14, 28 and 42 days after stress. And also the effective photochemical yield was also increased under stress. And while the photoprotective mechanism it shows the reducing trend, and the stress indicator FV over FM ratio, it also shows less reducing trend, which shows that more effect on the effect of photo inhibition. And in clustering, the clustering analysis suggests that our germplasm could be divided into four groups. Could be divided into four clusters. The first cluster, which had the properties of NPQ, while all the other physiological traits showed but doesn't contain the properties of all the other physiological traits. In case of second cluster and third cluster, they are quite opposite. Like in second cluster, it shows less values, low values FV over FM, but in cluster 3, it shows higher values of FV over FM. So we show that these genotypes are less affected by the stress, and they maintain their activity of photosynthesis. And here it can our germplasm variation, percentage of variation by PC1 was explained 65%, and by PC2 was almost 13%. And in correlation, all the physiological traits showed correlation, except we also have used this PAD chlorophyll index. It doesn't show any correlation under heat stress condition, while under control condition it shows only correlation with stomatal conductance and transpiration rates. So for drought screening experiments, I used polyethylene glycol 6000 as a drought inducer. We used 33.33 gram per liter of PEG to induce a drought pressure of minus 0.129 megapascal, which is almost equal to the field drought stress condition whenever plant has field drought stress. So in this we select the tubers, and then we when they attain the equal heights, then we transfer them to our hydroponic facility, and then we perform other analysis. In case of analysis in the drought screening, we again perform the from the IRGA, from which we again get net photosynthesis rates, stomatal conductance, transpiration rates, and CO2 assimilation. We also perform chlorophyll analysis of A, B, and carotenoids. And for assessing what is going on inside plant or leaves, we measure osmotic adjustment by using a vapor meter. And as we can't use air, we can't use chlorophyll fluorescence, so we replace it with the thermal imaging to assess the which we can get from pixel analysis. We can get the information which plants are showing better response to stress, or which are not, as you can see in these pictures. The other based on the other physiological parameters, we also tended to perform other antioxidant, other enzyme analysis like total soluble sugars, sucrose contents, proline contents, total soluble amino acid, and thiobarbituric acid reactive species, hydrogen peroxide, ascorbate, peroxidase, catalase, and peroxidase dimutase. All of these analyses are currently going on. So currently I don't have the data on this. So that's why I only list them here. The initial results from IRGA, and that's just us that the under control condition where we didn't apply any PEG, we only give the nutrient solutions. The commersonii was highest in net photosynthesis rate and also in stomatal conductance and along with the transpiration rates. While during when we applied PEG, then the chacoense was higher in photosynthesis rate, and stomatal conductance was very low, and also the transpiration rates. And while after removing stress, I called it a recovery phase. When we remove the stress and apply and allow them to recover for some days, in this case, we after like 10 days of recovery phase, we again measure the same traits, and dihaploid plants, tuberosum dihaploid, they can't able to survive the stress, so they are very like susceptible to drought. They died, and while the commersonii showed the highest recovery rates in case in all of these physiological traits. So in this, I showed the E of each species of each accession response to control drought and recovery. And here you can see that BGB 94 in chacoense in control and in stress condition, and then after recovery phase, they showed visibly they showed tolerance and recovers very fast. And also along with it 95, they also showed the same results in 96 and 97, and while 98 they recovered, but their appearance looks not as much good as the other accessions. And other remaining remaining like BGB 103 was one of the accession. Even during the stress condition, it doesn't show much susceptibility, although visibly it looked like very tolerant to drought, and it maintain its appearance, and even after recovery it looks like the same. So visibly we can say that the BGB 103 was one of the drought tolerant, but we need to look we need to justify this by other analysis like enzyme activities, so we can differentiate between all of the accessions. In case of commersonii, only these 6 accessions showed the better results. Like in 48, as you can see that under stress condition, the appearance of the 48, and after recovery it recovers very fast. The 55 session number 55, their recovery rate was very fast, and it shows a very great appearance results. And likewise, the other accession like BGB 77, 451, 453, and 460. One thing is I would like to mention here that all the commersonii we used here are from the collection from field from Brazil, and the chacoense we used are most of them are from introduced from the other stations, from USA and from Argentina and other breeding stations. In case of tuberosum, as I mentioned earlier, that they didn't able to survive, but only one session which able to survive and recovered very fast. It was the BGB 89, and all of these session like 88, 91, 93 died, but it last in the end. The one interesting thing about these sessions are that these are the only four sessions which tuberize first, and they tuberize first, and they are very really early maturing varieties accessions. So maybe so this is also interesting. They under stress they complete their cycle and produce tubers and die. And only this session it also produced tubers, but it stays green until the end of the cycle when we decided to harvest all of these sessions. So along with this all work, the coming up in future, we have a plan to do the genotyping by Jugas. For that, we are going to send our samples to Michigan State University for through which we there with this whole cap in phenium array, we are going to genotype our all of the sessions available in our germplasm bank, so we can get so we can differentiate between them and map them and get more information about them. Along in this project, the future our future plan is to screen all the sessions, available sessions in vitro also. And so for that we already have planned to in vitro apply. We already run some pilot projects, like I try to do on some of the sessions just to see the response, how we can manage in future this project. And then the tolerant ones, the genotype which will show the tolerance to heat and drought in vitro, we will move toward the proteomic analysis, like what will be the role of the what is the role of the HSPs in their tolerance. And also we will perform transcriptomics to see what genes are active and what biological processes are going on. Initially, we also had planned to do the hybridization, but due to some constraints, we can't able to do the hybridization. But in future, we after screening from in vitro, we have planned to move them towards hybridization. For that, we are going to we can use different methodologies, because most of the germplants we have here are diploid, and we have to hybridize it with the tetraploid. So whether either we will do it by chromosome doubling, or we will use bridge crosses. So this is all. In the end, I would like to acknowledge the fundings from the campus, PEPERS, TWAS, CNPQ, and the sessions provided from the USDA, and these for a dihaploid session from the breeding program of Dr. Caesar, and the research facilities provided from by Imbrapa, UFPEL. So thank you for listening me with patience. Now, if you have any questions.
Thank you so much, Ikram, for your speak. We're now open for questions. So you can either post them in the chat if you would like, or just raise your hand and open your audio and do it directly to Ikram. Pedro is congratulating you. Questions, anyone? Yes, Pat, do you want to go ahead and just ask Ikram directly?
Yeah, sure. Ikram, thanks for the talk. I noticed that your PC1 and your heat stress study was 65%. That seems pretty high. I'm not used to seeing numbers like that. Could you reflect on what you think that might mean a little bit?
I only show the show in the stress side, but in the control side, it was also surprising for me that in control side the PC1 was like 42, but while under stress condition it was 65, maybe because of the also the correlation, because under control condition among physiological traits there was not much correlation between the traits, while under stress condition a lot of the physiological traits like ETR, effective photochemical yield, and FV over FM, they showed a significant correlation. Maybe these are the reasons. I don't know if I able to answer your question.
No, that's great. Thank you.
Great, Pat. Thank you. We have another question here, actually two here in the chat, Ikram, from Max Feldman. The first one is, will these accessions be evaluated for drought tolerance in the field? Max, if you want to go ahead and ask him directly,
Sorry, I don't understand. Can you repeat?
Oh, so he's asking if you're going to test for a drought tolerance in the field or just in greenhouse.
So in field, these are, I don't think so we are going to do, because this is just like the start. This is the first time we are doing on this germplasm. So right now we are just focusing like in greenhouse in controlled conditions, and next time, next our plan is to do in vitro, and then maybe we will move toward the field. But right now, I don't think so.
Got it. Just as a follow-up, how many replicates did you use per accession? Is it just a single plant? Or do you have like, I don't know, groups of 3 or 5 or 10 plants per accession?
So in heat stress condition, for my first experiment, I used 3 accessions, 3 replicates, and in drought, I used 2, because of the availability of space, and also because of there is a mess of roots. So I have to be very limited so we can differentiate them.
I know how much work that is. Great job. Very nice presentation. Thank you.
Welcome.
Thank you, Max, for your question. We have lots of congrats here in the chat, Ikram. So everybody is congrating you. Does anyone has any further question you would like to go for, Ikram? So we have a question from Jonas in the chat, if you want to look at it. He says, the metabolite contents you perform in the tubers or leaves, if you did it, I suppose. And did you observe any correlation between the photosynthesis and the sugar levels? Have you measured the sugar levels?
I think I told in my presentation that first we are going first part of the question, when tubers or leaves, we are going to do in leaves, not in tubers, because in drought stress conditions, not all of these sessions tuberize. So we are only going to do in leaves here. And for the second one, I can't answer right now, because I'm still doing, and it is under process. So I don't know, maybe we'll find, maybe we don't.
Great, thank you, Jonas, for your question. Ikram is still open for questions if anyone want to ask him.
Sure, I'll ask another one. Ikram, that's a whole lot of phenotyping. What do you think you get the most value out of? What do you feel out of all the things that you measured? What are the most reliable? What are the most informative? And what are the easiest to take as far as the measurements go? Can you just talk a little bit about that?
Yes, I think
What's the hardest too?
I think the most reliable and the most easiest measurement which I able to do was for IRGA, like photosynthesis rates, and also for chlorophyll fluorescence. So based on these, if we find some differences, really significant differences, then we can, I decided to move toward the other metabolites, enzymes. So because they are all very time consuming and resource consuming. So in this way, I plan to move forward to explain more deep into what's going on in the plant.
Okay.
Great. Any further questions? Yes, Ikram, I just wanted to comment that I saw your pictures using the Leica, and I forgot who asked you if you're going to do that in the field, and Leica is a very expensive equipment, right? Piece of equipment, and going with it in the field is quite, well, it's not a nice feeling, you know. I'm just saying that we've done that in the forest and in the Amazon. I mean, it could be challenging. It could be very challenging. So you have all the limitations to do that in the field as well. Just want to say that. Oh, I think we have no questions, no further questions. So I would just like to end thanking you again, Ikram, for your participation in the Solanaceae seminars and for such a nice presentation. I wish you luck in your near future going on with this experiments, and I hope you got your PhD soon with these amazing results. Okay. Thank you so much, Rocio or Chelsea. You want to add something, or we're good to go?
Oh, I just wanted to add that next, in 2 weeks, we'll have the presentation of Robert Last, that he's going to present about from the tip of the trichome to the root of the matter, evolution of metabolic diversity in tomato and its relatives. And with that, that's all. We are finishing the seminar. And
if you missed, I mean, everybody's here. It's just being here, but if you know someone who missed, you know that you can watch later on YouTube, and it will be available really soon. Thank you so much.
Bye.
Bye-bye. Bye, Ikram. Thank you.
Bye. Thank you.